Good morning, everyone. Hi, my name is Jasmine. I'm with the Computer Science with Business Applications group in BCOE, and my project that I'll be discussing today is titled Using High Throughput Screening Techniques in Chlamydomonas reinhardti to Identify Genes that Promote High Levels of Lipid Accumulation. Okay, so as we know that global warming is an increasingly hot topic today, even though, as seen on the left, global emissions of CO2 have been on decline as a whole, global warming is still on track to reach 1.5 degrees Celsius Fahrenheit above the kind of standard before pre-industrial revolution, regardless of how much greenhouse gas emissions fall or rise or change in the coming decade, as seen by the proceedings of the National Academy of Sciences. So something we need essentially is something that's renewable, carbon dioxide neutral, something that's extremely productive. So what we found was that alkyl biofuels could fit all those needs. So algal biofuels have the potential to reduce greenhouse gases. They have a significantly smaller carbon footprint than traditional forms of energy. The CO2 release on production is consumed by the growing algae, so essentially it is carbon neutral. And when used in conjunction with a bioreactor, it is 400 times more efficient at removing atmospheric CO2. Additionally, the growing and cultivation of the algae does not interfere with the scarce resource of fresh water. It can be grown in wastewater, brackish water, or salt water. Yeah. And then so what we want to find is how this algae can be transformed into fuel that we can use. So the premise of this project was to find that the naturally produced lipids in the algae is produced through stressful environments. So what we found was that nitrogen deprivation kind of really enhanced that, and find the genes responsible for lipid production. So the lipid that we are looking for is triglyceride, or abbreviated as TAG, and it is naturally produced by algae, and it is easily converted into biofuel or liquid fuel. And to become an economical alternative, we need to increase the growth rate of algae and increase production to maintain consistently high levels of the triglyceride. So the algae that we are studying is the Chlamydomonas reinhardti. It is a unicellular haploid alga, and we chose this because it is well studied and well documented. Um, so the data that we used is this knockout mutant library provided by the Joincus lab. It has about 18,000 coding genes and 64,000 mutants in one library, and it covers about 83% of the nuclear protein coding genes. And so the knockout mutant library is where each mutant has at least one gene that's knocked out or turned off, so that we find that the relative genotype kind of is linked to a phenotype that we can observe, and which makes it likely that the gene that the phenotype that we observe is likely related to the gene that has been turned off. And each mutant has a barcode, and because each gene has multiple mutants, we want to find what mutants and what genes are responsible for the lipid production. So the algae was first dyed with this lipophilic dye, Nile red, which means that it tends to combine with lipids. And we used FACS, or fluorescent activated cell sorting, to identify the genes and sort them into like high lipid or low lipid or just like unsorted. So the FACS, or fluorescent activated cell sorting, is where the cells are sorted into like one stream thick, a stream of one cell thick. And as a stream passes through a laser, the laser fluoresces the Nile red. And if the fluorescence is high or low, it is then sorted into like high or low fluorescence. And as you can see on the left, that is what on top is what the low lipid cells look like, and the bottom is what the high lipid cells look like. So it is something that we have seen and is observable, and that is due to the fluorescence of the Nile red dye. So the mutant abundance is determined by the internal barcoding sequence of the mutant populations. The populations of high and all lipids, are high and all, were sorted of high and low and all of the algae were sorted and sequenced. the internal barcodes of the individual mutants were quantified to tell us how much of each mutant was present in each population. So as you can see on the left, that is an example of a barcode. That is the DNA that we are interested in. And as seen in the graphic below, in this high lipid gate, the pink mutant is clearly enriched in this gate because there are several more cells or algae compared to this green. And if you look at the all, you can see that the pink ones were sorted into this high lipid gate, but not all of the green ones were sorted into this high lipid gate. So because the pink mutant is enriched in this high lipid gate, it is likely that it has a high lipid phenotype that we want. So the part that I had a role in was the processing of all this data. So this already, the stuff I mentioned before was kind of already given by the chlamydomonas knockout library. And so what do we do with all this data? So first, we found the ratio of the high lipid gate versus the all inside each screen, and then found the p-value using the Fisher exact test and the adjusted p-value. So the p-value is like the probability of under the assumption that there is no effect of obtaining a result that's more extreme than what we actually observe. So if we want a low p-value, which means that there is a low chance that what we see is due to chance, and that gives us confidence in our results. And so we also adjusted this p-value using the Benjamini-Hochberg correction, which helps us avoid false positives due to stringent controls on the p-values. Since we have such a large data set, it helps us pick out mutants that we are interested in rather than just kind of taking this large bucket of genes or possible genes. And it kind of runs them through this more strict test to find actual important genes. And so our strategy after getting the adjusted p-value is bin each of the mutants and genes into these buckets. So since we have multiple mutants per genes, the way we organize the mutants in each gene was separating it by ratios. And if each gene has a unique combination of mutants organized in bins, that means that is significantly different than the population, then that gene is likely involved in lipid accumulation. So we have a histogram on the right, which kind of shows all of the p-values in this screen. And then on the right is how we sorted them into the percent bins. So here we see that about 40,000 genes or ratios made it into the 68%, and then another 51,000. So that's like the combination of this 40,000 in conjunction with like another 12,000 that makes it into this 80%. So that 12% difference is what is in this 80%, and then so on and so forth. And then when we get to like 97%, then we see that there's about a 9,000 difference or 11,000 like ratio difference, and that's what's in between the 17%. And so some of the results that we found is that this large data set has identified genes that are involved in lipid accumulation, which will help engineering strains for alkyl biofuels. And as seen as this heat map here, we see that for all of the runs or tests that we've done, we see, so this is like all the genes, and these are all the screens that were done earlier. And so the darker the cell is, the less likely that it is, or the higher the adjusted p-value. So what we really want is a really low p-value, adjusted p-value. So so then this reduces overall costs, or will help reduce overall costs and get one step closer to making algal biofuels a reality. So some of the challenges I encountered was that in the beginning I had this runtime of 6.5 days, which was kind of not as really unrealistic because this is an iterative process. So we need to run this over and over again with several different variables. And so the way I found out that I had this problem was that I realized I had kind of like a nested loop that just did redundant calculations over and over again. So the runtime of my analysis was directly related to the square of the size of the input. So we had thousands of mutants, so that kind of came out to about 6.5 days, which means that it took 6.5 days to come up with like one heat map. And then, so the improvements I made was that I entered the p-values into the adjusted p-values into a dictionary where the key was the total number of genes per bin, and then the value of that key was mapped to the value of adjusted p-values. And this I found was a linear runtime, and that kind of cut down the 6.5 days to 1 hour, which is much more reasonable and can be run over and over again. So I would like to thank Professor Jinkerson, my mentor and the principal investigator for this, and Elizabeth Hahn, who is also another mentor of mine. Thank you.
Thank you very much. We'll go ahead and move into questions. As a reminder, if you have questions, please go ahead and enter them into the chat. I actually have a couple questions to